Hi, I'm Chase, and I did the Gummy Bear Lab. This lab is a functioning experiment of osmosis. The normal water goes inside of the gummy bear and causes it to expand. The sugar molecules will try and spread out and disperse evenly throughout the bear. The gelatin membrane will not let them out, though, which causes it to get bigger. And for the salt part, the salt causes the gummy bear to shrink because the greater amount of salt in the salt water causes the water to exit the bear. The gummy bear will shrink more in a higher concentration of salt water. This is because there is more salt than anything in the bear when it is added to the water. Molecules pass through the semi-permeable membrane from a less concentrated solution into a more concentrated one as a way of stating this osmosis. And my questions are, how long will it take to expand a gummy full size? For question 1, which is regarding the water. And question 2 is, how long does it take to shrink a gummy to a certain size?
Hi, my name is Kaylin Baggett, and today I'm going to be reviewing receptor-mediated transport. This process is a form of endocytosis, meaning it is active transport and does require energy. The structures involved in this process are the plasma membrane, a coated vesicle, a coated pit, receptors, coat proteins known as clathrin, and a specific target molecule. The steps of the process are as follows. First, there is a plasma membrane that features a coated pit covered in receptors that are coded to sense a specific molecule. When these receptors collect enough of this molecule, they send a signal to the plasma membrane. This will result in the membrane forming around the molecules and becoming a coated vesicle. This vesicle then releases from the membrane and travels into the cell. My two questions are, what function do you think clathrin serves in this process? And then secondly, receptors can sometimes bring harmful molecules into the cell, such as the flu virus, diphtheria, and the cholera toxin. Why do you think that this is?
Diffusion versus osmosis. How are they different? Diffusion is the movement of molecules, solutes, from higher concentrated area to a lower concentrated area until equilibrium is reached. Osmosis is the movement of water, solvent, from higher concentrated area to a lower concentrated area through a semi-permeable membrane until equilibrium is reached. If you look at the pictures on the left, the first one shows more sugar molecules on the left side than the right. In the bottom picture, the sugar molecules move from left to right until the molecules are balanced throughout the cell, known as the state of equilibrium. This is diffusion. Looking at the right side, in the top picture, there are more sugar molecules on the left side and more water molecules on the right side. However, through the process of osmosis, as you can see in the bottom picture, water molecules move to the left side. The movement stops when the water concentration is equalized. Both are passive transports, meaning they do not require energy. Now moving on to the questions. Question number 1. What kind of transport is diffusion and osmosis? Question number 2. When does the movement of water through the semipermeable membrane stop during osmosis?
Hey, it's Kaylee, and I'm going to be talking about osmosis in red onion cells. So, osmosis happens to balance out the concentration of the water molecules on each side of the cell membrane. This is an act of passive transfer, and there is no energy required in this process. Shown for you are red onion cells with their cell membrane and their cytoplasm labeled. When you add salt solution to the cells, they will shrivel up or shrink. This is because water diffuses out of the cell to even out the water molecules on each side of the semi-permeable membrane, causing it to decrease in size. If you soak up the salt solution and then add distilled water, the cells will expand or swell. This is because water diffuses back into the cell, again evening out the water molecule concentration and increasing the size of the cell. And now I have some questions. Our first question is, if you add salt solution to a cell sample, will the cells swell or will they shrink? And our second question is osmosis an act of passive or active transfer, aka, is there energy required in this process? Okay, thanks. Have a nice day.
I'm doing osmosis in red onions. The quick definition of osmosis is particles in a solution that move from a highly concentrated area to a lower concentrated area through a selectively permeable membrane. In other words, it's just diffusion through a semi-permeable membrane. This is before salt water is added, so fresh water is added and the cells soak it up, making them expand. The solute concentration here is low, which makes this a hypertonic solution, and the higher concentration of water outside the cell makes the water flow into the cell. Here you can see the cytoplasm, the cell wall, cell membrane, and the nuclei. After salt water, the salt water is added and the cell structures in the cell start to shrivel. Since there is a higher solute concentration, which is salt in this case, would make this a hypertonic solution. The higher concentration of water is inside the cell, so the water will flow out of the cell. Passive transfer is when water is moving on opposite sides of a semi-permeable membrane and has the ability to move through the membrane. This is passive transfer, meaning it requires no energy. My questions to you are, is there a higher solute concentration before or after the salt water is added? Why? And why is osmosis a form of passive transfer? Thank you.